This video will aim to describe the structure of DNA, whilst also explaining how this structure enables DNA to code for proteins. First off, the DNA consists of a sugar phosphate backbone. This forms a double helix spiral structure. Across the middle, joining the sugar phosphate backbone, are bases. There are 4 different types of bases, joined together by weak hydrogen bonds. The 4 bases are adenine, thymine, guanine, and cytosine. Adenine always binds with thymine, and guanine always binds with cytosine. This is called complementary base pairing. A section of DNA is called a gene, and it is a gene that codes for a particular protein. Now let's have a look at how this structure of DNA enables proteins to be created. So looking at our DNA, first step is the DNA unravels. This enables some RNA nucleotides to come and bind with the exposed strand of DNA. It's the same idea as how DNA, the bases in DNA bind together. We've got 4 bases in RNA. You've got adenine, guanine, cytosine, but you've also got a new one, uracil. Uracil replaces thymine in RNA. So in DNA, we've got ATCG. In RNA, we've got A, G, C, and U, with U replacing T. Now we still get the complementary base pairing. Take a look at the diagram. We've got G with C, we've got A with T, however, we've now got U with A, we've also got C with G, and here we'll get another C with G binding there. Another difference between RNA and DNA is that the RNA molecule here is a single-stranded molecule. DNA is double-stranded. Okay, so to recap, 2 key differences between RNA and DNA. RNA is a single strand, DNA is a double strand. RNA has A, U, G, C. DNA has A, T, C, G. The aim of transcription is to produce a strand of messenger RNA. This strand of messenger RNA will then leave the nucleus and travel into the cytoplasm. The next stage of protein synthesis is called translation. So, the messenger RNA has left the nucleus via nuclear pores and it's travelled to the cytoplasm. In the cytoplasm it binds with a structure called a ribosome. These ribosomes hold the messenger RNA in place. During translation, a new type of RNA is used. It's called transfer RNA, or tRNA. At one end of the transfer RNA, we have an amino acid. At the other end of the transfer RNA, we have a sequence of 3 bases. Look at how the sequence of 3 bases will match up with the sequence of 3 bases on the messenger RNA. Once again, it's complementary base pairing. In this example, we've got AAG binding with UUC. It's still RNA, it's not DNA, so we've got those 4 bases that are found in RNA, AUGC. So transfer RNA brings in a specific amino acid. It's joined to the messenger RNA at the ribosome, and as new transfer RNAs bring in each amino acid, they join together. Eventually, we end up with a growing chain of amino acids, all joined together. We would call this chain of amino acids a polypeptide. Eventually, that polypeptide will fold into a certain shape and form a protein. Remember, proteins are made up of a chain of amino acids. So here's a picture to summarize the whole process of protein synthesis. Transcription is the first stage of protein synthesis. It takes place in the nucleus of the cell. The aim of transcription is to produce a single-stranded molecule of messenger RNA. This messenger RNA is similar in structure to DNA, however, it is single-stranded and contains uracil instead of thymine. This messenger RNA leaves the nucleus via nuclear pores and travels to the cytoplasm, where it binds to a ribosome. This stage is called translation, where transfer RNA brings in specific amino acids to the ribosome. The transfer RNA binds with the messenger RNA complementarily. As more and more transfer RNA molecules are brought in to the ribosome, amino acids are joined together, and eventually they form a chain of amino acids. A chain of amino acids is called a polypeptide. Eventually, this polypeptide will fold into a certain 3D shape and form a protein.